We're going to be learning about genetics. This chapter is all about chromosomes, DNA, inheritance, and about genetic disorders. I will first start with the definition of a chromosome. A chromosome is a long thread of DNA. So you could see the DNA double helix here. This is a section of DNA. So this is a gene. A gene, by definition, is a section of DNA that codes for a protein or a polypeptide. So DNA is not entirely made up of genes. Some sections are responsible for making and coding for proteins. These are the ones that are called genes. The whole length of DNA, after it's been coiled around some proteins called histones, that's what we call a chromosome. So chromosome is one long thread of double helix DNA. In this picture we see the definition of a gene. So right here we got the section, the gene. A gene codes for messenger RNA during the process of transcription, and in the next section messenger RNA will be translated into a polypeptide, and then this polypeptide will fold into a 3D protein. So you could see the whole events of protein synthesis here. Step number 1 is transcription. Step number 2 is translation. Human chromosomes and higher animals are always arranged in pairs. So for humans, every 2 chromosomes that are inherited from our parents are quite very similar. That's why we call those chromosomes that are similar but not really identical, we call those homologous chromosomes. So humans got 23 pairs of homologous chromosomes. The last pair here is what we call the sex pair of chromosomes. Those could be XX in females or XY in males. This is the only case when the homologous chromosomes might be significantly different. So it's only in males you will find that the homologous chromosomes are quite similar. But if you look everywhere else, the size, the length, and the banding, they are quite very similar. We're going to find out what are the similarities and what are the differences between homologous chromosomes. Here is an example. Now let's take a look at this homologous pair of chromosomes. Let's suppose that the gene for eye color is found here, and this person has received from one of his parents a brown eye color. Let's code for brown with B capital. And let's suppose that he has received blue eye color from his other parents, and let's code for this with B lowercase. So you could see that you might receive 2 different copies of the same gene. These copies will be different by some DNA bases. Few DNA bases will make all differences between brown eye color, blue eye color, or different blood groups. So this location right here, the location where you find the gene, always the same gene, will be found on the same position. This position is what is known as a locus. So a locus is the position of a gene on the chromosome. Chromosomes are quite very big, so that's why we have to find the position of the gene on the chromosome. And as you can see here, the genes could have different versions. These different versions of the gene, like brown and blue, these are what we call this alleles. So alleles are different versions of the same gene, which are always found at the same position or the same locus. both alleles will be expressed. So in this case, the B capital for brown and the B small for blue will both be expressed. However, there is always one allele that is stronger and masks the effect of the other allele. The allele that always gets expressed, even if you have one single copy of it, is known as the dominant allele. So dominant allele is the one that you only need one copy of it to have this expression and to have the appearance that is caused by this allele. We have a word for appearance. We call this the phenotype. So phenotype is the expression of the gene. It's always the dominant allele that is the one that is responsible for the phenotype. Recessive alleles like B small do not get expressed unless the dominant allele was absent. So the only way for a person, based on this example, to have blue eye color is to be B small, B small. So basically he has or she has to receive two identical copies of the B small allele. We are now going over the rules of inheritance to decide what are the phenotypes of the children or the offspring. Now, if one parent has pure dominant or homozygous dominant genotype, like B capital, then 100% of the offspring will have his or her phenotype. There's no way, if one parent was B capital, there's no way for the offspring to have any different phenotype, because any one of the offspring has to have at least one B capital that he has received from his dominant parent. The other rule is the 75% rule. In this case, if one parent was B capital, B small, that's known as heterozygous, and the other one is B capital, B small, then 75% of the offspring will have the dominant phenotype. So how do you prove this? You prove it by drawing the old Punnett square and put in the genotypes of the parents here. So you could see that every box of these will have a B capital. It's only the last box that will have a homozygous recessive, and that will be the offspring with the blue eye color. So again, if both parents are heterozygous, then the offspring will have 75% of this particular phenotype. The 50% rule states that if one parent was pure recessive or homozygous recessive and the other one is heterozygous, then 50% of the offspring will have the dominant phenotype. So you could prove this again by drawing a Punnett square. So one parent is heterozygous and the other parent is homozygous recessive, and you could see here that this offspring will be brown, so is this one here. The rest will always be blue. So that's the 50% rule. The last rule is you call it the 0% rule. That states that if both parents were pure recessive or both parents were homozygous recessive, then there's no way for the offspring to have brown eye color. There's no way for the offspring to express a dominant phenotype. We'll change the topic now and talk about mutations. Mutations fall into two major categories. Point mutations are the ones that involve the change of one base or one nucleotide. We always use this example, which is a sentence that is written in triplets. It's written in triplets because the genetic code, as you know, is a triplet code. Now, in this example, it states that you may change one base or one nucleotide on DNA without causing any harmful change to the organism. That's because in many cases, many triplets code for identical amino acids. So I'll take this example here. If the letter C in the original sentence was changed to K, now obviously this is written in a different spelling, but if you listen to the sentence, you wouldn't notice that there is any change. So this is an analogy that shows you that triplets like CAT or KAT will code or will mean the same amino acid. That's because the genetic code is a degenerate code. Degenerate code means many triplets code for the same amino acid. So that's something good, because mutations could occur without causing harm to the organism. Such mutations are known as silent mutations. Silent mutations do occur, but they don't change the type of amino acid, and they won't cause any harm for the organism because they're not going to change the 3D structure of the protein. In other cases, point mutations could be really harmful. They could be harmful if the change in the triplet causes the formation of a new acid. So if we change the C into, let's say, B, for instance, then this triplet will now mean or will code for a different amino acids. Sometimes if the amino acid has the same nature, then that won't cause a serious problem to the 3D structure of the protein. But if you change an amino acid, let's say that is hydrophobic, with an amino acid that is totally different nature, let's say if it's charged or hydrophilic, then that will cause damage to the protein structure, and the protein may not fold in its right way. Point mutations could be even more harmful if one base was changed with another. So let's say this G guanine has been swapped with an adenine. Now, changing in this way, this means that this ATT, this particular triplet, will now code for a stop codon. A stop codon is the one found on messenger RNA and will result in an early ending of the process of translation. That's because this will be transcribed into a UAA, and UAA is one of the 3 stop codons. So if this mutation occurs early in the gene, means like at the beginning of the gene, then the protein translation will stop very early. It is going to be less harmful if this stop codon was formed near the end of the gene. So again, point mutations where that code for a stop codons will be less harmful to some extent if they are near the end or near the actual stop codon. There are 2 other types of mutations other than point mutation. One type of mutation is called deletion. A deletion is the one that removes or deletes a base on DNA. So let's say that this C was deleted. This will cause what is known as a frame shift. A frame shift stands for the fact that every single triplet after the deleted base will now be read in a different way. So this is going to become ATE, this will be TET, and you could see now the entire sentence is unreadable because of the loss of one base. Again, frame shifts are going to be less harmful if they occur later on or near the end of the gene, but nonetheless it will cause damage and effect on the entire triplets that come after. The last type of mutation is known as insertion. and insertion is the addition of a base, it will have more or less the same effect of deletion, because it will also cause a frame shift, meaning that it will affect all of the triplets that come after. Here we have a summary of three types of mutations. So the three types are point mutations, insertion, and deletion. Now we've seen earlier that both insertion and deletion cause more damage and effect than point mutations. Point mutations are quite harmful if they lead to the formation of an early stop codon. Nonetheless, point mutations could change one amino acid, but there also could be a silent point mutation if the triplet that has been changed codes for the same amino acid, or codes for an amino acid of the same nature, meaning if the amino acid was charged and the mutation leads to the formation of another charged amino acid, then it won't cause much damage. We're gonna look here on how a mutation, point mutation in this case, will cause a damage to the 3D structure of a protein. So mutations occur on the level of DNA. Mutations don't occur to RNAs. Mutations could occur by the change of, let's say the C here has been changed to a G, and this triplet is now different. Now this change in the DNA will cause a change in messenger. So the messenger now, instead of this guanine here being guanine, it will now become cytosine, and the triplet now will code for a different amino acid. So during the process of translation, you will end up having a different amino acid. A change in one amino acid could cause a change in the 3D structure of the protein. So this is a model protein. This is actually insulin. So if this person was unlucky and the amino acid that was mutated was one responsible for maintaining the 3D structure of the protein, then this whole protein won't fold and it won't take its 3D structure. Another example, if the protein to be translated was an enzyme and the change in this amino acid that was mutated was part of the active site of the enzyme, then this enzyme won't fit its substrate and will be turning a non-functional protein or enzyme. We are now going to talk about genetic disorders. One very well studied disorder is cystic fibrosis. Cystic fibrosis is a recessive disorder. This means that for a person to have this disease, she must be pure recessive or homozygous recessive. Cystic fibrosis has major effects on the respiratory system. It also damages the digestive and reproductive systems. Cystic fibrosis has to do with one particular carrier protein. This carrier protein is found in all parts of the body that are responsible for secretion of mucus. In this picture, we could see this carrier. It's known as CFTR chloride carrier. It's responsible for the transport of Cl negative ions from inside the epithelial cells into the mucus. So chloride ions will pass along these channels into the mucus, and you could see that the ciliated cells nearby are the ones responsible for pushing the mucus away. Mucus's job is to trap bacteria and harmful material, so it's quite important to keep this airway clean by cleaning up the mucus with the help of cilia. So again, the mucus's job is to trap the bacteria and the ciliated cells are to push the mucus. As we've seen earlier, genetic mutations occur on the level of DNA. So a change in the genetic code will cause a change in messenger RNA because messenger RNA is transcribed from the DNA. So any error in the DNA is going to be copied. In the case of cystic fibrosis, the change results in the formation of a different amino acid, and this results in the formation of a different carrier protein during the process of translation. So CFTR, which is the carrier protein of chloride ions, becomes blocked due to this genetic mutation in the DNA. A blocked CFTR prevents the transport of chloride ions. Chloride ions usually move around from the inside of the cells, so that's the inside, to the outside where the mucus is found. In all cases, the chloride ions being mixed with the mucus cause the movement of another ion. So this side becomes very negative, and it helps the movement of another positive ion, which is sodium. This presence of both chloride and sodium within the mucus drops the water potential of the mucus, which means that water from the inside of the cell will move down a water potential gradient and will mix with the mucus. This actually increases the fluidity of mucus and helps the mucus to become more watery or more fluid so that it could be put with the ciliated cells. So the ciliated cells here can push the mucus away because it's quite fluid. In the case of cystic fibrosis, the chloride ions cannot move and water will be retained inside the cells. This results in very thick mucus. The ciliated cells in this case cannot push the mucus, and this results in too much mucus being blocked inside the airways and inside many channels within the bodies, including the pancreatic channel and sperm ducts and other channels that contain mucus. Having mucus been trapped means that bacteria will also get trapped inside the airways. This results in many infections related to bacteria that could occur inside the lungs, inside the pancreatic channels, or any channel where mucus is found. In this picture, we see that too much mucus inside the airways will actually reduce the flow of air and oxygen to the alveoli. So in this picture, you see that the alveoli are receiving less air and less oxygen, plus the distance between the alveoli and the capillaries around them becomes more, and that reduces the rate of diffusion of gases in these cystic fibrosis patients. So right here we see that the lack of air getting to the alveoli and the increase of distance between the air inside the alveoli and the red blood cells found in the capillaries nearby both contribute to the lack of gaseous exchange. That's why patients of cystic fibrosis tend to run out of breath, and they are usually struggling with tiredness. They're usually tired because they lack respiration. They're not respiring as normal people, and they cannot release the energy that is needed for their bodies. We're gonna work here on scenarios of different questions related to cystic fibrosis. So questions will ask you to try to find a link between cystic fibrosis, a mutation that leads to cystic fibrosis, and certain symptoms in patients of this disease. So for instance, if you've been asked what does the mutation of cystic fibrosis has to do with symptoms of tiredness and breathlessness. So the way you could answer such a question is by trying this track. So first of all, you start with the mutation blocks the CFTR channel. This channel, the CFTR, is responsible for the transport of both chloride and sodium. Now, the lack of sodium and chloride in the mucus makes it become way too thick. So the thick mucus results in the lack of water in the mucus. Having a thick mucus causes two consequences, one of which is blocked airways like the trachea and the bronchi, and this results in lack of oxygen and lack of respiration. Now, respiration is the way we release energy, so there will be less energy released, and with less energy, that will explain the patients being always tired, and that explains why they are breathless. They are breathless because they try to breathe faster than usual, trying to compensate for the lack of oxygen delivered to their blood. Another symptom related to the thick mucus is the blocked pancreatic channel. You know, like the pancreas is an organ where enzymes are made. The blockage of the pancreatic channel, meaning that those patients will have less digestive enzymes and will suffer of digestive problem. We're going to talk now about sex-linked genetic disorders. A sex-linked genetic disorder is the one that is more likely to be found in one gender than the other. So for instance, color blindness is a genetic disorder related to sex or gender, because males are more likely to be colorblind. So for instance, if you're a male and you're having difficulties seeing the number in this picture, this means you're having this disorder which is known as color blindness. The reason why males are more likely to be colorblind than female, that's because the allele for color blindness is found on the X chromosome. So that's the X chromosome, and the color blindness allele is found here. So if you are a male, then you will only have one copy of the color vision allele, and you won't have a backup copy like the way females do. Here is an example of how color blindness can occur in a family where both parents are quite healthy. So for instance, the father here is not colorblind, so when I write the genotype of this father, I will write it like XB capital, meaning that he's healthy, and I will leave the Y chromosome blank because the allele is only found on the X chromosome. The mother here is a carrier, so when I write her genotype, I'll write it like B capital and B small. This means that she has the allele for color blindness, but she doesn't show a sign of this disease. When I am to predict the genotype of their offspring, I'll draw the Punnett square, and you could see that the first possibility here is that the mom or the little daughter here will be healthy. You could have another daughter that is a carrier like her mom. The boys could be either healthy, like this one here, or could be a colorblind boy. So 1 out of 4, or 25% of the children are likely to be colorblind if the mom was a carrier and the dad was healthy. Another genetic disorder that is related to sex is hemophilia. Hemophilia is a disorder that is also carried, just like colorblind, it is carried on the X chromosome. In the case of hemophilia, the XH subscript here stands for the allele that causes this disorder, where patients are more likely to bleed, and this capital H stands for a healthy blood clotting. We have possibilities here. Now, if the male has this genotype, this means he will struggle because it takes too long for his blood to clot. Only females can be carriers because females can have two copies. One is the healthy one and one is the mutated one, which is lowercase h. Females are very unlikely to be hemophilic because the chance here is 1 out of 3. It's very rare to find a female that is suffering of this disorder. Usually males are way more likely to be having this genotype, which is X, H small, and Y. Just like the way we've done it with cystic fibrosis, you can always prove that parents that are healthy, like those here, can have a child who suffers of hemophilia. And as always with sex-linked disorders, males are the ones that are likely to have the disorder. So in this case, if you try to breed those two parents, then you'll end up having a girl that is healthy, another girl that is a carrier just like the mom, a boy that is healthy, and a boy that struggles or suffers with hemophilia. So in this case, just like the way we've done it with cystic fibrosis, there's a 25% chance of having this disease.